everybody, myself Ramesh. Especially today we will talk about very general topic, especially in Kjeldahl method, the estimation of protein. So you can see the estimation of protein nowadays. There is many methods to estimation of protein from organic or inorganic substance. You can see the most frequently used methods for measuring protein content in food, including the Kjeldahl method, UMass method, direct measurement methods using UV spectroscopy and refractive index measurement. Especially in Kjeldahl method, it involves like mainly 3 steps. You can see the first one is the digestion of organic matter with sulfuric acid. That is the first one point is very important. And the catalyst they are using, they are making the reaction produce in alkali. Then after distillation and titration to label the ammonia. And then you calculate the content of ammonia. Then you got that result, you multiply these with 6.25 to get the CP content, means crude protein. So crude protein over the evaluate the amount of a protein in milk is because it includes non-protein also. So especially you can see here, first you can see the introduction of Kjeldahl method. So Kjeldahl method was first introduced to existing in 1883. One of the scientists in chemistry is developed Johann Kjeldahl. Johann Kjeldahl is the first he introduced in 1883. The method was specially developed by the determine the nitrogen content in organic, especially in inorganic compounds. So the methodology is first developed to determine the percentage of nitrogen in organic and inorganic substance. So nowadays today the world Kjeldahl method nitrogen determination are used in several samples like wastewater, soil, fertilizer, meat, feed, grains and many other substances. Nowadays they are using Kjeldahl method to estimation of protein. So this method is specially used for the estimation of protein content in food. Now you can see what is the principle of Kjeldahl method. This is very important. See, this principle if you understand, then you will be easily known about the procedure and this method. So digestion of the test portion with concentration sulfuric acid. So that is the first point. And using the copper sulfate as the catalyst to convert the organic nitrogen to ammonium ion. That point is very important. This alkalization, distillation of the liberated ammonia into an excess of boric acid solution. So the titration with hydrochloric acid to determine the ammonia bound by the boric acid. So the calculate of the nitrogen content of the sample from the amount of the ammonia is produced. Actually that is the principle. The first principle is, you can see here, the digestion of the first portion with concentration of sulfuric acid. That is the first point. And that heat you can maintain in by the adding the copper sulfate is the one of the catalyst. Then that catalyst will make the organic nitrogen convert into ammonium ion. That is the point. Those ammonium ion is trapped in a boric acid solution. So the titration with hydrochloric acid, then you can see how much ammonia was released from this our substance. Then you can see, before we are going to discuss about that, what is the crude protein and what is the true protein, that you need to know. So what is the true protein? Nitrogen compound is only has the protein, that is true protein. What is the crude protein? All nitrogen compounds including also non-protein nitrogen, NPN, in the addition of protein, that is called crude protein. So NPN means what? Non-protein nitrogen, the like free amino acids, anemia, like ammonia, urea, burate, nitrate. These are called as a non-protein nitrogen. Then you can see what are the apparatus to why you are doing the Kjeldahl method. So Kjeldahl method mainly the 3 steps. One is the digestion unit. So digestion tubes are needed, and distillation operator means digestion tubes, conical flask and distilled water, and then titration unit, titrated standard and conical flask. These are the 3 main parts operators are needs to make the Kjeldahl method for estimation of protein. Then you can see this is that digestion unit and this is the distillation unit. You can see clearly, we will discuss in next slide. And you can see the somewhat closer of the what is distillation. You can see here stood that surface of here by heating coil, Inspection of windows, here is how these samples, and acid fumes protecting surface, here is, this is acid surface protecting, and lift junction is here, and you can see this is the menu, operating menu, and renewable control unit here, and insulator handles to carry it out upside, and this is the circuit breaker, and this luminance. This is the somewhat closer if you see the distillation kit or distillation unit. Then you can see here, then you can see the turbotherm. Turbotherm is one of that digestion infrared heating system. So you can see this is that carrying of this part is called turbotherm to use as the heating system of this digestion part. So you can see this is the one digestion, this is the distillation. You can see here. So the first one is the, you can talk about the first step is the digestion. What is digestion? You can see here, this is our sample. Then you can add the powder like one catalyst, potassium for sulfate, then is the copper sulfate, another one the catalyst, there both are catalyst using, and to add the sulfuric acid H2SO4. So this is that reaction mechanism. Our sample is organic compounds, nitrogen, sulfuric acid to produce carbon dioxide, water and ammonium sulfate. And these ammonium sulfate use, well, the going to the digestion, then after you will move on to the next step is the distillation. So distillation, you add the sodium hydroxides of this liquid especially, and then the color was come in reddish, pink or brownish color, and liberated the ammonia in the sample. And this is the part of distillation you will carry it out, and this is the mechanism, the reaction mechanism. Then the final step is the titration. So the clear green and the colorless solution, you can observe your solution. Then after you can see the methyl red, methyl blue red indicator. Then you can see this is the actually end point of the paint pink color. You can see this is the titration with H2SO4. You can see the further. So this is the application of Pelican's equipment, the Kjeldahl nitrogen analyzer or protein analyzer, both they can use. And I can see here another one, the steam distillation and digestion. These are special units of this Kjeldahl method. Then you can see here, this especially in digestion method, you can see this is the digestion tube, and here is our ammonia trapper, and these are the, here you can see initially the boric acid will pass, then after sodium hydroxide, then after you can go for the water. This is actually the distillation unit for the Kjeldahl method, and this is the one of the object. What is the actual object of the Kjeldahl method? The qualitative determination of organic as well as the inorganic nitrogen in the substance. So first one is the digestion, and step 2 is the distillation, and step 3 is the titration. This is the 3 main steps for the Kjeldahl method, and this is the objective of the Kjeldahl method for the estimation of protein or the estimation of nitrogen. Then you can see somewhat closer what is digestion. For the decomposition of nitrogen sample utilizing the concentrated sulfuric acid and catalyst is copper sulfate and K2SO4 for boiling point increasing agent. This catalyst is the main role to increasing the boiling point. So increasing this boiling point of the catalyst, and as the result of ammonium sulfate is produced. So this is the equation. You can see nitrogenous compound copper sulfate and the H2SO4 sulfuric acid K2SO4, then this is that ammonium sulfate produce water plus carbon dioxide and other byproduct. This is the actual equation. The process was done in digestion. Then you can see the distillation. By adding the excess base of the acids, digestion mixture is converted into ammonium to NH3, NH4 to NH3 to followed by the boiling and condensation of the ammonium gas as received in the solution. This is the ammonium sulfate, and you add the sodium hydroxide to release the ammonia NH3, and sodium sulfate plus water will come byproduct. And the final step is the titration to quantify the amount of ammonia receiving in the solution. By the back titration we will do in the final step. Then you can see what are the reagents. Sulfuric acid using for has one of the reagent, and catalyst, you know, the 9 parts of the potassium sulfate anhydrous and nitrogen free with 1 part of copper sulfate CuSO4, and the sodium hydroxide 40% in dissolving sodium hydroxide in technical grade for the distilled water. Then boric acid 4%, anti-bumping granules you need to use, and the ethanol for using for the 95% standard for the titration using the sulfuric acid or the standard sulfuric acid, then indicator they are using methyl red and the bromocresol for the green color. These are the actually stock solution and reagent while you are doing the Kjeldahl method you need to prepare. Then you can see here the overall what is that means the overall of Kjeldahl method is divided into the three main steps. One is the carried out the proper sequence. The step is digestion, distillation and titration. So this is the main equation. Our sample and copper sulfate and the potassium sulfate and 350 degrees Celsius heat, then ammonium sulfate will be releasing. Then you can see the for more details you can see digestion, distillation and titration. You can see somewhat closer. You can see in this method, especially in digestion, the substance sample is heated with the sulfuric acid. So and this sulfuric acid break down the organic substance via the oxidation and reducing the nitrogen into the ammonium sulfate is liberated. The potassium sulfate is usually added to increasing the boiling point. In the medium, catalysts like mercury, selenium, copper, this copper also used for the digestion process. This sample is fully decomposed when we obtain in the clear or colorless solution. You can see the organic compound for a sulfuric acid to digest in copper plus ammonium sulfate. Then you can see the distillation what is happening. The distillation of solution now takes place in the small quantity of the sodium hydroxide added to the converted to ammonium salt or to ammonia. The distillation vapors are the trapped into a special trapping solution of HCl, hydrochloric acid or water. This is that equation. Then titration, the amount of ammonia or amount of nitrogen present in the sample is determined by the back titration. So as the ammonium dissolved in the acid trapping solution, some of the hydrochloric acid has neutralized. So acid that is left behind this can back titration to standard solution base such as the sodium hydroxide or other bases. This is the equation you can see for the titration. Then you can see here next step. And next step we can see why they are using the boric acid used in Kjeldahl method. That is nice question. So the boric acid captured the ammonia gas forming the ammonium boro complex. So actually that is that boric acid main purpose is to trapping the ammonia, like ammonium borate complex has the ammonia collecting in a color of the receiving solution changing. So the boric acid method has advantage that only to standard solution of necessary for the determination and it is used solution for the long shelf life. And then why the limitation? What is the limitation of this Kjeldahl method? So this Kjeldahl method is not work out for the all solution. You can see all substance or the all feeds or all food items. You can see this method used only for the nitrogen bounded of organic compounds. What organic compounds are bound with nitrogen? Suppose you can see protein, amino acids and nucleic acids. These are the ammonium in the sample. So those is you can determine by the Kjeldahl method. So other than this method is not suitable for this compound. This point is very important. Nitrogen like azo compound, nitroso group of in rings, quinone, pyridine, nitrate, nitrite, etc. These kind of we can't estimate from the Kjeldahl method. This is the one of the drawback for the Kjeldahl method for estimation of protein. And you can see how this calculation was done. This is equivalent of ammonium into 17 and you can see 14 grams of nitrogen is containing one equivalent weight of the ammonia. So weight of the nitrogen is soil in digested in these formulas. You can see another one formula, percentage of nitrogen volume of H2SO4 by number of H2SO4 into 14 by weight of the original sample weight into 100. So crude protein is equal to nitrogen into conversion factor. And this is the calculation of crude protein. You can see using of this formula you can find out for the crude protein content. And you can see the expression of results. The method of calculation is formula nitrogen content in sample used for the using of this formula, and you can see the total protein is equal to nitrogen into 6.25. Actually this is if the addition of result in terms of nitrogen. Suppose if you need to nitrogen and resort to the express in the result in protein, so then you can use for this formula n into 6.25. This is a factors. This is that value to